Hello everyone. Today we are going to discuss about the DNA, the genetic material, and which is the transforming material from one organism to another organism. What are the things we are going to see in this particular topic is what are the requirements to be a genetic material and the experiments conducted by the Griffith, Oswald T. Avery, McLeod and McCarthy to prove that the DNA is a genetic material which transform from one bacteria to another bacteria. What are the requirements of the genetic material is the first character is it must carry the information from one generation to another generation, and also it must replicate by their own enzyme mechanisms, and it must carry the information to change according to the time to time, and it must govern the expression of the phenotype. And what are the key characteristics of the genetic material is it must contain the complex information, it need to replicate faithfully, encode the phenotype, have the capacity to carry the information from one generation to another generation. And there were discussion about the protein, RNA and DNA in these three different components which were considered to be a genetic material. And the lots of experiments have been conducted to suggest that DNA is the genetic material rather than the protein and ribonucleic acid. And the first experiment was conducted by Frederick Griffith in the year of 1928. He is a British bacteriologist and his main focus was on the epidemiology and pathology of bacterial pneumonia. The organism named the Diplococcus pneumonia, later it was called as Streptococcus pneumonia, which have the capacity to cause the infection known as pneumonia. And the experiment on the bacterial transformation, it was first supported to prove that the DNA is a genetic material of bacteria in the year of 1928. And he transferred the genetic properties from one strain to a second strain, and he has taken the two different forms of the bacteria. One is the R form organism and second one is the S form organism. R form is known as the rough colony producing Streptococcus pneumonia, and S form of the organism is called a smooth colony forming Streptococcus pneumonia. And the R form, they are avirulent strain and it does not have the capsule, and when it is grown in a media they produce the rough colonies. And the S form of the organism, it is a virulent and bears a capsule which makes the organism to produce or it may cause infection to the individual. And in this right side you can see the picture which represent the colony characteristics in the media, the organism which possesses the capsule. The first setup is he had taken the R form cells of Streptococcus pneumonia and he injected in the mouse, and he found that the mouse was live. And the second treatment, he had taken the live S form of the organism of Streptococcus pneumonia and he injected to the mouse, and he found that it caused a pneumonia like infection, and as a result it caused the death of the animal. In the treatment 3, he had taken the S form of organism and he heat killed them and he injected the mouse. Then he found that the mouse was survived and it was live. In the fourth setup, he has taken the heat kill form of S form of organism and he mixed with live R form organism of Streptococcus pneumonia. After mixing it, he injected to the mouse and he found that it caused pneumonia. As a result, the mouse was found to dead. And in the next, we are going to see the animation about the Griffith experiment.
Have you ever wondered what determines your height, hair and skin color, and even the size of your nose? It's DNA, the genetic material of cell. But do you know how it has been proved? Here is the experiments that proved it. Griffith's experiment. Griffith took 2 strains of pneumococcus bacteria, S3, which have smooth polysaccharide capsule, giving smooth colonies S, and it's virulent, causing pneumonia, and R2, which have no capsule, giving rough colonies R and is avirulent, unable to cause pneumonia. When the virulent S3 bacteria injected in mice, it killed them, causing pneumonia. But when the avirulent R2 injected in mice, the mice lives. If you injected the heat-killed virulent S3 bacteria, the mice still live, implying that live S3 bacteria are needed for causing pneumonia. But interestingly, when mice were injected with the mixture of heat-killed virulent S3 bacteria and avirulent R2 bacteria, mice died. Analysis of dead mice reveals live S3 virulent bacteria, suggesting something from the heat-killed S3 had transformed to the live avirulent R2 bacteria, responsible for the transformation of the live avirulent bacteria to virulent S3 bacteria, causing death of mice. This is called as bacterial transformation. To prove DNA is genetic material, team of Avery et al. And Hershey and the Chase performed another.
And next we are going to discuss about the Oswald T. Avery and his companion. In the year 1944, they set to discover which constituent in the heat-kill pneumococcus was responsible for Griffith constitution. And they had taken the heat-kill S form of organism and they extracted, and by using the saline soluble components, and they centrifuge those components and they separated and extracted the DNA, RNA and protein molecule. And what they did is they had taken each and every component and they combined with the different enzymes which destroyed each component separately. And they had taken the extract from the S-type cell and they mixed with DNase enzyme, and after the treatment they mixed that particular heat kill S form of the extract which is treated with the DNase and they mixed with the R-type cell and they inoculated into a media. Next they found that it produced the R-type colonies. It means that the rough type colonies. And the extract of the heat kill S-type cell, it was treated with the RNase enzyme and they mixed with the R-type cell and they inoculated in the media. On next day they found that the R form of the organism have been converted to produce the smooth type colonies on the media. And the third setup, they had taken the extract of the S type of cell, they treated with the protease enzyme which degrade the protein molecule which is present in that extract, and they mixed that particular extract with the R-type cell and later they inoculated into the media. On next day they found that the R form of the organism have been converted into smooth colony producing organism. It means that they have acquired a new character, that instead of producing the rough colonies they produced the smooth type colonies. It means that they got a new character to produce the capsule, and by this experimental setup they proved that the DNA as a genetic material was mixed with the RNase enzyme and the protease. When they mixed with the R form of organism, they found that in both the setup or both the components which treated with the RNase and protease enzyme, it have produced and converted the R form of organism into S type of organism. And this is the experimental setup to prove that which component is considered to be an hereditary material. And they had taken the heat kill S form of organism and they extracted by centrifugation mechanism, and they had taken each and every component in different test tube and they treated with the proteinases and ribonucleases and deoxyribonucleases, and these organism then it was mixed with R form organism, and they found that the R form organism which is treated with both the proteinases and ribonucleases were found to convert into S form of organism. With this they found that the transformation mechanism have been carried out. The component from the S form of heat kill organism have transformed into R form of organism to convert them into the S form of organism. It means that it have converted to a virulent strain and they got a new character to produce the capsule. The component which is treated with the deoxyribonuclease enzyme, what happened is it have denatured the DNA molecule, and when they mixed with R form of cell and when they inoculated into media, they found that no S cells were appeared, and by this setup and with these results, they proved that the DNA is the transforming material and it is considered to be a genetic material which have carried the information from one generation to another generation. And the third setup or the experiment which was conducted by McLeod and McCarthy in the year of 1944, and they have extracted the components of S type, it means that the smooth type of cells of pneumococci by centrifugation, and it is being the virulent strain, and they have used the different components. One is the enzyme which degrade the protein, and second one the enzyme which degrade the DNA molecule, and the setup was done separately, and they mixed with the R-type cell after treating with the enzyme that degrade the protein and enzyme that degrading the DNA, and astonishingly they found that the enzyme which is degraded with the protein, when it's mixed with the R-form cell, it had transformed the R-type cell into S-type cell. It means that it has produced the smooth colonies because of getting the new character to produce the capsule. When the enzyme that is degrading the DNA, when they mix with R-type cell, they found that it have produced only the R-type cell in that media. Next, we are going to see a small animation and video about the Avery, McLeod and McCarthy experiment.
Hello everyone, quick biochemistry basics here. Let's talk about Avery McLeod McCarthy experiment. Streptococcus pneumonia has two types of strain, smooth strain and the rough strain. The smooth strain have a protective capsule, as a result of which they are highly virulent, whereas the rough strain is not virulent. In the year 1928, Griffith discovered that when heat-killed smooth strain is mixed with the rough strain and injected in mice, the mice died. This result was surprising. It means something was getting transferred from heat-killed S strain to live R strain that made the rough strain virulent. This phenomenon is known as the transforming principle. Griffith did not know what was getting transferred from heat-killed S to live R strain. So in the year 1944, Avery McLeod McCarthy did a series of experiments to find the transforming principle. They took cell lysate of heat-killed S strain and treated with different enzymes. Once the respective enzyme treatment is given, the mixture is treated with R strain and incubated, and finally spread it on Petri plates to see what happened to the rough strain. The presence of smooth colonies means that the rough strain got converted to smooth strain. It was only DNA's treatment that failed to convert the rough strain to the smooth strain. This means that the DNA was the transforming principle.
Already we had discussed that the DNA is a genetic material, and also the experiment conducted by the Griffith, Avery, McLeod and McCarthy to prove that the DNA is the genetic material and which is considered to be a transforming material which transforms from one organism to another organism. And this is the source for the reference of this particular topic. In the next session, we are going to discuss about the RNA as a genetic material and the experiment which is conducted to prove the RNA is a genetic material by Conrad and Singer in the year of 1957, and the Alfred D. Hershey and Martha Chase in the T4 bacteriophage to prove that the RNA is a genetic material in the year of 1952.
Turning the clock back to the 1940s and 50s, the scientific community was in the midst of a debate about the nature of the hereditary material that is responsible for passing our traits on to our children, to our children's children, and so on for generations. Pretty existential question, right? An experiment by Avery, McLeod, and McCarthy in 1944 first suggested that proteins were not responsible for heredity, as previously thought, and instead DNA was the culprit. However, given that their results flew in the face of the current thinking, not everyone was convinced. In science, a great deal of evidence is needed before a new scientific idea is accepted as fact. Therefore, in 1952, Alfred Hershey and Martha Chase carried out experiments which addressed a similar question. Was DNA the hereditary material, or was it protein, or was it something else? To solve this important puzzle, Hershey and Chase needed a simple model organism to solve this complex question, and a clever experiment. They chose the simplest organism possible, a bacteriophage, which is a virus that infects bacteria. This virus looks a bit like a lunar landing module and attaches to the outside of the bacteria. It then injects a substance into the bacteria to create many new copies of the virus. These newly manufactured viruses are released when the bacteria bursts. So the substance injected inside of the bacteria must contain the instruction manual for making new virus. It contains the hereditary material. But what was this material that the virus injected into its host? The answer to their question might shed light on the nature of heredity for all life on Earth. Viruses consist of two things: a DNA core surrounded by a protein coat. Hershey and Chase needed to figure out which of the two was injected into the bacteria. They ran 2 experiments, and in each they labeled one of the components. For this labeling, they used radioactive tags: radioactive sulfur for the protein and radioactive phosphate for the DNA. This was an elegant method because phosphate is only found in DNA and not in the amino acids that make up proteins, and sulfur is only found in proteins and not DNA. After the viruses infected the bacteria, the researchers examined whether the radioactive signal was detected inside the bacteria or left outside. To do so, they needed to separate the bacteria from the viruses attached to their outsides, and did so using a Waring blender, similar to that used for making milkshakes. Once separated, they then centrifuged the sample. Since bacteria are bigger, they were centrifuged to the bottom, while the smaller viruses remained at the top of the tube. When they prepared virus with sulfur-labeled protein and mixed it with bacteria, they found that most of the radioactivity remained with the virus and did not enter the bacteria. When they allowed the virus to replicate and collected the new viruses, less than 1% of the protein radioactivity from the viral parents were found in the newly replicated virus, so the protein was not being inherited. On the other hand, when they performed the same experiment using virus with its DNA labeled with phosphate, they found that most of the radioactive DNA was injected into the bacteria from the attacking virus, and much of this labeled DNA was inherited and passed on to the new virus generation. Hershey and Chase added to the mounting evidence, pointing the finger at DNA as the hereditary material, but they were aware that their experiments left a few unanswered questions, as experiments often do, and they were cautious in their conclusions. From their observations, they stated that DNA had some function in the multiplication of the virus, whereas proteins did not. Word about the Hershey-Chase experiment spread quickly and helped to convince many scientists that DNA indeed was the molecule of heredity, and the remaining skeptics could not hold out for much longer. A year later, Watson and Crick published their model of the DNA double helix, which provided an elegant explanation for how DNA with such a simple structure could be replicated and pass along information from parents to offspring. The Hershey-Chase experiment is considered a classic in biology because it tackled an important question with the use of a well-chosen model organism and an elegant experimental design in order to clearly distinguish between 2 competing hypotheses.